Hi, I'm David Taylor, and I'm a professor at the University of Texas at Austin. I study structural biology, and today I'm going to talk to you about why being a scientist can be so cool and very different from the stereotypes that you might see in TV shows and other forms of media. It can actually be quite exciting and fun. So we're all familiar with the picture of the cell that we get from an introductory biology textbook, which is this circular object, and occasionally it has this circle in the middle, which stands for the nucleus. And in the nucleus, all of our genetic information is encoded, which allows us to function as an organism. So in reality, the situation is much more complex. So here I'm showing you two images of the crowded cellular environment that's composed of all of these little nanomachines that are performing functions. So on the left is the cytoskeleton. So motor proteins move along the cytoskeleton, transporting different nutrients, signals, and neurons. They're also important for the plasticity and shapes of the cell. On the right is the nucleus. And so in the nucleus, you can see that there's these helical bundles. These are actually 30 nanometer fibers that contain nucleosomes. And in the nucleosomes, our DNA is wrapped around it. It contains all of our genetic information. You can see some purple molecules. Those purple little shapes are actually polymerases, and they're either replicating the DNA so that the cell can divide, or they're transcribing the DNA, creating messenger RNAs that are later expressed into proteins, and that's part of the molecular biology, so central dogma of molecular biology. So here's a gallery of macromolecular machines. And so when I say macromolecular machines, I mean these little nanomachines that do all of the work in our cells that allow us to survive. And so you can see in this image, they come in various shapes and sizes, and they do various things, from viruses that infect cells to polymerases that, like we talked about before, replicate or transcribe DNA molecules. And so my lab really focuses on understanding the structure and function of these machines. A lot of times by getting a picture of them, by understanding the structure, we can understand how they work. So the mantra for my lab, and I think really science in general, is the closer that we look, the more we can actually see and understand about our environment. And so on the very left, you have an image of the McDonald Observatory, also a part of the University of Texas at Austin. And so there you're looking at things that are very far away. So universes, planets, stars. In the middle, you're looking at colliders. And so those are where we use high energy to bombard particles together and break them apart to create subatomic particles and study them. And so on the very right is the Titan Krios electron microscope. And those are the microscopes that I use. It's a very big microscope to study very small things, so these nano machines. And to give you a scale for what these machines actually look like that we use the microscopes themselves to study the nanomachines. Here is a picture of one of my graduate students next to the microscope. So we use these gigantic microscopes to study these very tiny things. Okay, and to explain a little bit about the science that I do, I'm interested in gene editing. So that's where we use enzymes called CRISPR or CRISPR-Cas9, which you probably heard a lot about in the media recently, to find a gene of interest and replace it. And so we can use this to cure diseases. And so here's an example that I'm showing you. So here's a chromosome with a flower colored gene. It has this DNA sequence that's composed of G's, A's, T's, and C's. Those base pair with each other to create a double helix of our DNA. So that's our gene. And that gene encodes for a protein, and this protein makes pigments. And in this particular case, it encodes for a purple pigment that creates purple flowers. And so then that's the final phenotype. So we're going from genotype to phenotype. So now we can easily read this code for humans. So with the advent of the human genome in 2001, we can now easily read genomes on a very fast time scale. But what we really want to be able to do is to take that information from the genome and use that to edit it and cure diseases. And so how can we do that? We use this amazing fantastic enzyme called CRISPR. Okay, and to just give you an example, I'm going to use a word processor scenario. So here I have a sentence that says, he gave me a glass of juice. That's great. I need orange juice during this COVID-19 epidemic to keep my immune system healthy. If I start to change the meaning, so if I start to edit the sentence or edit the genes, things can go awry very quickly. So here I'm getting rid of the word glass, so I'm editing the genes a little bit, and now I have a sentence that says, he gave me a of juice. It no longer makes any sense. If we made this edit, we would have a non-functional gene that could be very detrimental to me as an organism if I can't express, say, a essential gene in my body. Okay, let's look at a find and replace example that what CRISPR would actually do. So here it says, he gave me a poisonous glass of juice. That's not very nice. That's not what I want to do. So we want to find the poisonous part of the gene and replace it with something that's good and hopefully cure that sentence and cure that disease. And so we find poisonous and we replace it with something that's good, like a delicious glass of juice. So now I'm happy, everything's good, we're good to go. And so here's the structure of the protein called Cas9, this CRISPR protein that actually can do this find and replace mechanism. In white is the protein, and what it does is it interacts with the DNA, and so the DNA helix is in light blue and dark blue, so those are the two different strands that are interwoven to create, that's how most of our genes are situated in the genome. And so you can see there's this orange colored RNA, this is called the guide RNA, that's within the protein itself, which gives it the sequence specificity. So that tells it what gene to actually target. And so it guides it to the target gene, it unwinds those two pieces of DNA, and then it can create a double strand break. So it can cleave the DNA on both strands, and then we can replace that gene with the good gene that we want to place in there. And so it's actually like a scalpel, so it creates this very precise cut so that we don't have off-target effects, we don't do bad things to our body. And it's very different from, for instance, the scissors we used at elementary school to cut construction paper, where we got these really jagged edges. It's also very different from a chainsaw. We wouldn't want to take a chainsaw to our genes and cut it up non-specifically and very aggressively, because that would also probably cause problems for us as an organism. And so that's what I study. I hope that today I convinced you that being a scientist can be cool and that you should pursue a career in science. There's different avenues and paths that you can follow, but it can be fun and exciting. And it's very different from the textbook science that you are familiar with, and more akin to science fiction novels that you might read. We're really on the forefront of this wave of the scientific revolution, and I encourage you to join it. Thanks for listening to me.